Thank you everyone for joining. This is the National Honeybee Survey webinar. In attendance, we have participants of the National Honeybee Survey, Apiary Inspectors of America, representatives from USDA APHIS. We also have our friends at the Bee Informed Partnership. So thank you all for coming. I really am excited for what's to come. Today, I'm very excited to host Dr. Dennis vanEngelsdorp, who is professor of entomology at the University of Maryland. For more than a decade, Dennis has studied honeybees and specifically an epidemiological approach to studying disease. And today he's going to speak to us about resistance found in Varroa. So without further ado, Dennis, I would like to pass this over to you. So I will stop sharing my screen and enable it so you can.
Okay, great. Thank you, Rachel.
You're welcome.
Oh, I think you still have to enable it maybe.
Okay, I will do that.
Sorry, we practiced this yesterday, so we know we can do it. Yeah.
All right, you should be able to.
Oh, yeah, there we
Let
go.
me know if you can.
All right. Thank you again. It's a real pleasure to give a talk to this group. I mean, Apiary Inspectors of America, as Mike just so kindly reminded me, I was an apiary inspector for a long time. It's how I got to know American beekeeping, and I really sort of did inspire a lot of the survey ideas that we see implemented in the Bee Informed Partnership, and I think it's a real testimony to a lot of what we're doing. I'm really hoping now that I'm not as involved with Bee Informed Partnership to actually look at some of the history of beekeeping and history of bee disease in the country, and I think that there's a lot of exciting and interesting stories to tell about both American history and can teach us something about managing bees today too, I think, in some of that history. So hopefully you'll invite me again for a different topic. But today's topic, of course, is about Varroa and resistance and resistance management. And here I'm going to use the word resistance in a very broad context. Usually we think about resistance as a genetic resistance to a certain chemical. But here, as I'll explain later on, I'm going to use a much broader context. But I have to admit that when I was asked to give a talk about Varroa, I was sort of like, oh, you know. And I'm sure a lot of you spend a lot of time giving Varroa talks. And we give Varroa talks and we give Varroa talks. And it's sort of like, it's endless, right? These giving Varroa talks, Varroa are important and how important that is. But again, I was reminded that it takes time for new ideas to get spread. And so here, I love this graph and the fact that if we look at radios were invented somewhere like in the 1920s, but it took 1950s for all the houses in the country to have radios. That cell phones were much quicker. It only took 10 or 12 years for cell phones to sort of saturate people. But let's remind ourselves that most beekeepers we serve or help or try to help are backyard beekeepers. And very few of them stay in the hobby for more than 3 or 5 years. And so we have this really high turnover rate. And so we're always having to educate and figure out different ways of reaching this new group of beekeepers. And if beekeeping is, in fact, a communal act where we have, you know, your neighbor's bees affect you, then we sort of have to really try to figure out how we can make sure we're communicating to these new beekeepers so they're getting that information as quickly as possible. Rachel, I have a whole bunch of like waiting list things. Am I supposed to let them in or are you doing that? Sorry for the interruption.
I'll let them in, no worries. You can ignore those.
Okay, all right. The other thing about giving Varroa talks is that increasingly Varroa talks are lose-lose, right? You can go in and you can say, yes, you need to control Varroa, and here's how, and you can talk about all the different products that are registered. And there are people who are going to really give you a lot of stinky eye, right? And then the other extreme, you go in and you say, okay, here's a pattern of no chemical control for Varroa, all natural Darwinian beekeeper. And there's a lot of other people who are going to give you a stinky eye. And it's true that there's no way that you can give these talks and not make someone upset. And so we actually, with the Bee Informed Partnership and some of that work that Natalie helped lead in terms of doing surveys for on beekeepers, we worked with some sociologists, and we actually codified or tried to describe the beekeeping community. And I bet it would come to no surprise for any of you to find out that there are 30% of beekeepers who believe there are no advantages to treating colonies for Varroa. We call these the treatment skeptics. We also have 41% of beekeepers on the other extreme. These are the treatment adherers, and they see no disadvantages to treating. And so what's core to this, though, is that if you try, the sociologists try to figure out what the belief system is that treatment skeptics truly believe that they should minimize contact with the bees, that they should have as little to do with the bees as they can. Where treatment adherents believed that they needed to actively manage hives to keep them alive. So in a lot of ways, the treatment skeptics are like the bee havers of old times. You know, before the 80s, before mites, we talked about the bee havers. Well, the treatment skeptics are still of that sort of mindset. We can keep bees without touching. Now, I want to emphasize that when I'm saying treatment skeptics, that doesn't mean they didn't treat, and we'll talk about that in a minute. But they would prefer not to treat if they could, as most of us would, but they really don't see advantages to treating. But the good thing is both of those extreme groups believe the same thing. They think that we want to keep colonies alive, and they just believe that their way of doing it is the way to do it. So at least there's common ground there. People want to keep colonies alive, and that's good. And I think we can build from that point. It's also important to remember that 46%, most beekeepers, believe in a balanced approach. That you shouldn't treat if you don't need to, and there's an equal number of advantages and disadvantages to treating for Varroa, which is this balanced approach. So that's the good news. There is that middle ground, but we still have these polarized extremes. And certainly as we develop messaging for these different groups of people who have these beliefs, we have to sort of consider how we can code our message to make sure we're reaching different audiences. I mean, there's also a group of people who still don't believe Varroa are a problem. And I think that you guys in the Apiary Inspectors of America and with the National Honeybee Disease Survey, we have clearly showed, to my astonishment, how much Varroa is a problem. Here you can quickly see, I'm sure you've seen this many times. This is every year till the end of 2018 that we did the national survey. And you can look at the Varroa mite levels per month on average. This is 5 mites per 100 here. We now believe that if colonies have more than 5 mites per 100, even if you treat them, they're going to have severe losses. We think that 3 mites per 100, it's approaching 3 mites per 100, that if you treat even at 3 mites per 100, there are likely going to be problems. And I'll explain some of those problems in a minute here. But so you can see that there's just a large number on average. There's a large number of colonies that are simply above threshold. If we condense all those years together and just look at them by month, what you can see is that after August, if we look at July, mites are above on average threshold until nearly December. And so 65% to 70% of colonies in October are above threshold. And so that clearly has to be contributing to these high loss rates. And beekeepers themselves acknowledge it. It's one of the most, if not the most, it is one of the most continuously cited reason for high losses or unexpectedly high losses. And when we look at Natalie's work with the survey, and we look at the management practices and how they tie to losses, and then our elaborate system of developing things, we can condense some of those systems down, and we can look at just Varroa. And so in this graph, and I realize you can't read it, but I think that you'll get the picture very quickly. What we see are these 4 graphs are for 4 different groups of beekeepers. At the bottom here, we have the beekeepers who only use Varroa and non-chemical product only, and no Varroa control at all. And this is northern beekeepers that are small scale, southern beekeepers small scale, large migratory beekeepers, large beekeepers that are not migratory. What you can see right away is that the beekeepers who use Varroa only, with the exception of large migratory beekeepers, lost the fewest mites, if not the same number of mites, than if they used Varroa and non-chemicals. But it was the commercial migratory beekeepers that lost fewest if they combined treatments. And so this to me, because they're at the forefront of some of these Varroa resistance problems, this to me suggests that there are problems of real traditional resistance in terms of chemical resistance. And we all know that that's true, and we'll talk about that in a minute. But before I want to do that, it's really, so this treatment issue, have we really documented that treatments are? We know that the labels for these different treatments are pretty complicated. Are these treatments because beekeepers are applying them right? What's going on behind that? And I think that Kelly Kahelnuck's work with BIP. She was a PhD student in my lab and now is working at Washington State with Brandon on migratory beekeeping issues. She'd be a real great speaker for this group if you ever want her. But she helped do the Sentinel Apiary project with BIP for the last 3 years. And what she did was as you all know, the Sentinel Apiary program is where beekeepers send in samples once a month, and then we send out reports back to them and they see where their mite levels are. And this is exactly what we would have expected to see. At the beginning of the study, we expected to say, okay, May, your mites double every month on average. There's going to be this peak sort of in August. Then you're going to treat, and all their mite levels are going to go way down, and they're going to stay down. That's what we were expecting. But in fact, when she monitored all these colonies, all the beekeepers in that study, well over 100 different beekeepers per year, she could look at when they treated. And you can see here, so there are 6 of this group, 21 here. There's 21 here that 6 treated in May and June. Oh, sorry, let me say that again. You can see these different months. These are people who treated. However, only those in the green did the treatment actually reduce mite levels the following month. So about 30% of the colonies that were treated with a miticide had reduced mite levels the following month. That means that 70% of treated colonies had mite levels that were the same or more than they were before you started treating them. Now that's a serious finding. And so, well, what could be causing that? And of course, one of the reasons could be as well, maybe they're not treating the entire apiary. Maybe it's only part of the apiary they're treating, or maybe they're using products that aren't very effective. And so Kelly went to ask these questions of the data as well. And here what you can see is these line graphs. This orange line, and this will be true for the next several slides, is the no treatment. So that's the line for the no post-treatment load. So if you had a high load up here, you had a high load up here. That's what you expect. So that slope is people who did treat 100% or less than 100%, their lines, while the mite level still increased, you couldn't tell the difference between those who treated everything and only part of everything, but we could tell that they did treat because the mite population didn't grow as fast as if you had treated, if you hadn't treated at all, I mean. It's weird to give these talks without seeing whether they understand you or not. So I'm trusting there'll be some flag waving if you guys are needing some instruction. The other thing is well, did treatments work? And if you look at that, again, this is no treatment, and then these are all the different treatments. We'll parse them out. Basically, yes, treating is better than not treating, but it was very hard to differentiate between the different treatments. And so she spelled that out. It was only amatraz. You can see amatraz here where we saw a trend to decrease, if not a stable mite population from the beginning to the end, between months, from the start of between after treatment and then after treatment here. So you can see the green line. So there was a big difference there. But for the other ones, we had, so you can't really, oh yeah, here we see it. There we have coumaphos here, formic acid, thymol, and oxalic acid. And you can see that all of those decreased the rate of mite reproduction, but still the mite reproduction was increasing. So we also made a collaboration with Dr. Joel Gonzalez Cabrera in Spain, and he's a world authority on mutations that govern the region that fluvalinate interacts with. So he's a pyrethroid. He looks at Varroa mites, and he looks at that site where pyrethroid resistance gets initiated because of a change in the genetic code. And so we sent him all the mites that we could from all the different states, collected it from our national survey or from your national survey, our national survey, in 2016 and 17, and 17 and 18. And he compared that to resistance in different places in the world. The first thing you'll notice is there's this green mutation. There can be up to about 5 different mutations at this one site. This green mutation occurs both in the US and in Europe. This yellow mutation only occurs in the US. This blue mutation, and that's just the different substitution that occurred, only occurs in Europe. But this red mutation was only thought to belong in North America, but in fact now it's been found in Crete and in Greece, and they think that this was because of an immigration. Just emphasizing again our need to make sure we're disallowing the import and export of bees and mites when we can. We think that only happened in the last 3 or 4 years. But he also looked then at the mites across the country for those 2 years, in 2016 and 2017. You can see that there's a little bit of variation, but not a lot. On average, about 37.8% of the mites, or the colonies in the country, had mites that had the gene that would mean that they would be phenotypically resistant to fluvalinate or pyrethroid. And that was, we also were able to follow operations over 2 years. So the same operation was sampled 2 years in a row, and we could send those mites. And what you can see here is that they're paired. So the 2016, 2017, that's one operation, and so on. And I tried to illustrate here with these arrows that there's a downward graph. That means that there was a big increase. So that means that there's a big increase in resistance to the miticide, so that there's decreased effectiveness of the miticide. So you can see that here, and that occurred in the majority of times that there was, so it looks like they had more dominated their population. This is what we expect. We saw a couple where there was no change, but we did also see a couple where resistance got diluted. And in fact, the good news out of the Spanish work shows that if you don't treat a population of mites for more than 3 years or 2 years with fluvalinate, so after the first year and certainly after the second year, quickly, if they do have no exposure at all, they become susceptible to fluvalinate again. And that's because the gene that confers resistance is not good for the mite. It's better than dying from the pesticide, but it's not good for the mite in the absence of the pesticide. And so it means that those mites aren't as fit or don't seem as fit and sort of peter out. And so it seems that susceptibility comes back into the population quickly. I will say that Christina in our lab too, Christina Cristamone is in our lab too, and she is right now analyzing samples that come in from the National Honeybee Disease Survey for resistance to different products. Now, traditionally, we've used the PETIS test to do this, of course, but there's some sort of train of thought that we need to do this in a much more standardized way. So she's working with Steve Cook at the USDA and some other labs who've just sort of pioneered this work. We're using vial assays to more accurately measure the susceptibility and resistance to individual mites to different products. And so I imagine that by January, she'll have some results to share with you in that regard. An interesting thing that she's also working on is measuring mites. And we did this first out of a regulatory concern because we found some really small mites and we thought they were, well, those mites might not be Varroa destructor. They might be these other mites, you know, Japsiconi or some of these other Varroa mites that we don't think we have. But in fact, they've always ended up being Varroa destructor, even some very small mites. But we did notice that there was a big variation in size. And one of the many studies that Christina has been working on has been measuring mites that die after exposure to different chemicals. And we think we're beginning to see trends where smaller mites are sometimes more susceptible or resistant to pesticide treatments. And so that's the work, she's collected a lot of mites, she's done a lot of tests, and I think we'll see some results there. But the preliminary test showed evidence that mite size may influence how effective a product is. So there might be different populations of mites with different sizes, and they may respond to products differently. The other one is the messy one that we all suspect, and that's this idea of horizontal transmission of mites across the landscape. And again, I'm going to go back to Kelly's work where she did some really, I think, novel and exciting work in terms of practical stuff we can take back to beekeepers. But in this first study I wanted to explain is that these are different apiaries. So each one of these dots is an apiary. This yellow dot in the middle is the donor apiary. And that donor apiary had 4 hives in it, or more than 4 hives, but 4 hives of import. 2 had heavy mites, and she painted every bee in those mites red, colonies red. And then there were 2 colonies that did not have many mites, and she painted all the bees in those colonies blue. And then she put out all these possible donor apiaries within 2 kilometers of this original site. And here what you can see is the number of bees that she found at the entrances of these different colonies in these different apiaries. So what you can see right away is that it was not, as we thought, a lot of high mite bees that were visiting, but we did see a lot of visitors across the landscape. And as we expected, the closer the apiary was to the donor site, the more visitor bees you got. But she also looked at mites. So she could look at the mite population at the colonies that these bees visited. And the first thing you can see is that were they visited by a bee that was from a high mite donor colony? No or yes. You can see there's a trend there, but it's not mathematically or statistically significant. However, if you look at any donor visit, so did this bee get a visitor at all? Yes. Then the mite population increased much dramatically, more dramatically at that distant colony. So it looks like that bees, colonies that get visited a lot, have much higher increases in mite levels. So they're obviously getting mites dispersed. But an interesting part of that study, and here I think we have a lot of work to do, and it could really maybe help a lot of beekeepers. So we have to really think about how to get this word out in a very careful way, making sure we're not overstating what we think might be, is the use of robbing screens. Now, robbing screens, to me, I would use these as moving screens. Basically what they are is this screen that sits out front of the colony, so an incoming bee isn't going to notice, is going to try to get through the screen because they can smell the entrance over there and everything like that. But the bees are actually inside that colony, are trained to do this exit here. So they're all going out this exit. So this is a robbing screen. It's really good to prevent robbing come in. Also as a moving screen, it's great for ventilation, etc. So she put these on colonies in those donor apiaries, and look at these results. Colonies that had robbing screens on them lost much, or had much lower mite reproduction than the ones that did not have robbing screens. And so this opens up a whole bunch of questions, right? Like, what should you treat though? How long do you put robbing screens on for? And all these things. And we don't know any of those answers. This was just for a short period of time. But certainly I think that that's something we need to look at, and we should think about collaborating ways of getting this test out there. Because if this is true, this may help a lot of that unexpected jump in mites we see once in a while. So in summary, mites continue to be a problem. We need to communicate beekeepers from a common ground. We have to look for that common ground and meet them where they are. And we have to also think of those barriers to management and treatment effectiveness. There's genetic resistance out there. Should we be monitoring for that? Should we be thinking about community ways of like, okay, everyone in this region treat with this product this year and then don't treat for 3 years? Are there things like that we should be thinking about? There's maybe physical resistance, like in size, and how do we monitor that and give recommendations? And then, of course, there's this horizontal transmission. So you treat, but it's your neighbor's mites that are coming back in and causing you trouble. So, of course, there's thank you. Project Apis M has helped support with some of Christina's grant. USDA has funded a lot through APHIS, of course, the National Honeybee Disease Survey, and in cooperation with you, the Apiary Inspectors of America. Also, USDA ARS has helped, or NIFA has helped fund BIP and my lab to do several of these projects. So we should say thank you to there. So I think that was my talk.
Thank you very much, Dennis. Does anyone have any questions that they would like to bring for the speaker? I will unmute everyone. Let's see here. Okay.
Everybody's unmuted.
Oh, sorry. All right. Yes, feel free to ask your questions now. Again, if you aren't comfortable with speaking on the microphone, drop your question into the chat and I will bring it up to Dr. vanEngelsdorp. Okay. So go ahead. Anyone who wants to ask a question, go ahead and unmute yourself and ask your question.
I have a question about this time they treated, or on that one, they didn't have any treatments in February through April. Some of my beekeepers will do a strip of Apivar in February. So does she ever have the beekeepers do that? Do any of them treat in the wintertime? Because you gotta have some southern beekeepers on that survey.
So
The one
so
chart, you know.
you're talking about those charts where we monitored the mite levels over the 6 months.
Yeah.
Yeah. So there, yeah, beekeepers did whatever they did. So if they were in the southern states, they would treat whenever they wanted. And so that's why you could see when I showed you those first bars, those were people who treated at different times. And so when they treated in that time, if they treated during that time, you could see the ones that had a proper treatment and didn't have a treatment. What I didn't show, and I think that that's what you're asking, and that's a good question, but I didn't make that part of this talk, was, well, what happened to those colonies in the long run? Like, did they survive or did they not? And so you did track that, but that wasn't the point I was trying to make with that data, was that only 30% of applications, no matter when you do it across the course of the year, actually reduce mite load. So there's obviously difficulty. But there are, of course, different ways and different patterns that you can use to control mites. And one of Kelly's projects, and I would encourage you to have her talk, was to test one of the sort of suggested methods that came out of the big survey work. So she did what average beekeeper did and what the best practices were. And she found after 3 years, much greater success.
Thank you. There's actually a question. Here we go. So Joey Caputo in Utah asks, is there a hypothesis why formic acid is not as effective in reducing the growth of mite populations?
So increasingly, I think you're seeing that at different times of year, the products are working differently. With the formic acid, one of the things, I mean, the question is, well, how are you defining it not working? Often when you hear someone talk about formic acid not working, you're talking about a month later, like you go a month later and you check. And it's true for some of these organics or these acids like formic, you can go back a month later and find that the mite levels have increased. One of the reasons we think this is, and we have some photographic evidence of this at least, is that when you think about a mite, it has as these palps up front, and they have these sensory hairs that hang out there, and that's how they smell. And so when they are on a nurse bee and they jump into the cell, they smell the larva and they smell, is it about to pupate? And if it is, they stay there, and if it's not, they jump on the next nurse bee and go back out. When you treat with formic acid, you can clearly see that you burn off a lot of these hairs. And so it could be that what's happening is that the mite population has increased because none of the mites are invading the cells anymore because they can't smell the right stage. So I know that I've done some studies where you can clearly see that 2 months later, the mite population has seriously gone down after formic. And so sometimes I think it's about how we measure. But again, there's a lot of strange things happening with formic. It's one of the things Christina is looking at to see if the formic is affected by mite size as well.
And piggybacking off that question, is there a difference in effectiveness that you've seen between 1 pad and 2 pads? So between partial treatment and full treatment?
I wouldn't know that information. I don't know if Natalie's here and if she's actually looked at that in the data. So I don't have an answer for that. I don't know. There's a lot of tech transfer team members here, and you guys are much more close to this than I. So if any of you want to say something.
I apologize. Could you repeat the question, please?
I think Rachel, the question is there a difference in effectiveness with 1 or 2 pads of the formic acid?
Yes, and its effectiveness of controlling Varroa mites.
Right. I think someone just wrote a comment that the company that makes the formic acid recommends 2 pads. So that's a good thing to know. Yeah.
This is Blaine White. There's a nuance to that. The label clearly states you can either do 1 pad for 10 days, followed by a second 1 pad for 10 days, or you can do 2 pads for 14 days. Both treatments are in there. They're both full label treatments.
And does anyone have a sense of which one you'd recommend or you're using or people are using in your area?
Blaine, again, I've seen and I've used both. I tend to do 1 because it's easier on the queens. But I've seen plenty of people that are using 2 with no problems, and I've done it myself. So it partly depends on what your schedule is in terms of when you can return.
So I'm just looking at the chat, and several people are commenting. Great comment, I think, is do not apply 2 formic pads in the summer. So again, that's going to affect your queens. So it seems like you have to, there's like a fine line, of course, with any treatment on whether, if it's going to affect your bees. But it seems that formic acid can be particularly tricky using both pads in the summer.
Yeah, remember, this is Blaine. I'm in Minnesota where our temperatures don't always get that high. The other trick is to freeze the pads before you put them on, which slows the release that first day and gives you a little bit of a buffer. It also gives you much lower exposure to the formic acid as the applicator.
Okay, we have another question in the chat. This is from Samantha Auger. Looking for clarification on the experimental design of the horizontal transmission study. For the bees that were marked, were those presumably bees from the neighboring apiaries that were robbing the hives located at the donor apiary? And then those bees were observed back at their home apiary. Secondly, is this study published yet?
Well, so a good question. Kelly is certainly trying to get this published. She finished in May, and I know that this is in the pipeline. I will say that we thought too it would be robbing screens, right? It was robbing that was driving these higher mite loads. We don't think it's robbing anymore. We think it's snooping. And I think, I mean, if you're really interested in this, and I think it's worth having Kelly really outline that experiment really carefully, because there's some really interesting nuanced findings there. And also she can present the best management practices that she's come up with. I think she's given a BIP seminar on those. And I think, are those recorded? I think they're recorded, so you can get those as well. Because I think that that's valuable information. So it's a good point. I'd get Kelly to clarify, but no, I thought it was robbing too, but we don't think it's robbing anymore.
Yeah, thank you. I am definitely going to reach out to Kelly to see if she'll speak, because I agree. I think she'll be a great speaker for this group. Okay, so we have another question from Kim Scrim of Massachusetts. How about comparing off-label amitraz use and label amitraz use related to mite resistance? Does labeled products of 3.33% versus off-label products of 12.5% show more mite resistance and higher AI?
So it's a really good question. And of course, one way to answer that is, well, that's a really difficult thing to do because we can't go and test these illegal products. However, that's where I think Steve Cook and a whole bunch of other people, and Christina and I was joined them, has come up with a clever workaround for that. They're no longer using the 3.3% strips and cutting them up as you do for the PETIS test. They're actually coating the inside of the vial with the active ingredient. And so by that way, you're able to measure the lethal concentration of, and come down to the lethal concentration of the mites more precisely. I mean, you all know resistance theory as well as I do, right? Like it's only so far that you can dump more product on. I mean, so I'm imagining that your question is it a good idea to be putting 12% on the colonies? And is that going to drive resistance? Theory suggests strongly that it would. Yeah.
Thank you. Let's see. Does anyone have any other questions? Kim says thank you. Okay. How about mite resistance and hive size? Any work comparing the number of boxes on hive and mite resistance? So does hive size appear to have any effect on mite resistance? So overall colony size.
I think you're asking treatment efficacy, right? Not mite resistance is, I think, the question. I'm assuming. And so I think that, yeah, I think that that's part of the balancing act. If it's treatment efficacy, of course, you need more product to get out to more bees if the colonies are big. And I think that's why you're seeing some products. And I think there's going to be clarity, more clarity about this in the next year. I think Steve Cook's lab is working on this, is that some products work better at different times of the year. And I think the BIP people would suggest that too. And so does that have to do with colony size? That's quite possible. Does it have to do with the dynamics? That's quite possible. So I think there's a lot of questions there, but absolutely, I think mite size is going to have or colony size is going to. Do I know experiments that are experimenting on that directly? No, but I think that that's part of the complexity for sure.
Kathleen Prowl asks, would Apistan work if someone has not used it for several years?
Well, so the Spanish study would suggest that's true. It would also suggest that you weren't in contact with anyone else who had mites that were resistant. And so I don't know how isolated you were, but theoretically, an isolated population of mites without pressure from pyrethroids becomes susceptible again rather quickly.
Joey Caputo asks, what is the theoretical framework for robbing screens reducing horizontal transmission?
So so that, I mean, I think that right now we're a little bit surprised at how dramatic this was, because we thought it was robbing, and we don't think it's robbing. So that was our first idea, was, well, it's just robbing bees, or it's bees from collapsing colonies just moving anywhere they can. So we thought we would see a lot more bees from high mite colonies entering those if it was collapsing, or we thought we would see, we weren't expecting to see this snooping behavior. So I mean, I am just guessing. We're talking about theories. I mean, I'm interested. What is your theory? Why would this be? If it, I mean, we can all, I think we're all really good at this. Why can that be? You can put it in your chat. You can put the answer. So I don't know. I think, have we bred more permissive bees? It would be interesting to look at bees in Europe, because European bees have been bred not to rob so frantically. I don't know that we put a lot of breeding pressure not to have snooping bees. And so, or do we have more permissive bees that, sure, come on in, we don't mind. Like, you know what I mean? Like, they're not very guarding the entrance. I mean, there's a bunch of different explanations possible, but I don't know. I'd love to hear some ideas.
I also have a questions from Judy Wu Smart. She says, "Hi Dennis, nice to see you. How much of the mite resistance is driven by residues accumulating in the comb? Does stopping fluvalinate also require comb replacement? And what's persistence of these residues in comb?" And I apologize for my dog whining in the background.
No. So yeah, Judy, that's a good question. And of course, I mean, we have been sending in samples now for a long time. We've got a really good handle on the pollen stuff. I'm glad to say that I put in some, that paper is now in submission, but certainly you can see a lot of stuff in pollen. We've also now been taking wax for a long time, and we are indeed seeing a lot of residues in wax. So what does that have to do with maintaining resistance? It's a good question. I mean, there's been some talk too, have we bred bees that are just more pesticide tolerant because they're always in these toxic environments? So some really good questions. Yeah, people need to write a lot of grants, answer all these questions. So I mean, it's a mess. I mean, mite size, how much pesticides are in the wax, what you used last year, what type of mites you have, what genes they have, how big they are. No wonder it's a confusion.
Thank you. Hannah Whitehead asks, "What exactly do you mean by snooping behavior in the horizontal transmission study?" So can you just define what snooping means?
Well, you know how the bees go and they like sitting there and they just hover at the entrance and they sort of sit there and they hang around a bit? I mean, maybe that has a different technical name. Judy, you'd know. What's the technical name for snooping behavior? I don't know. But you guys all know what I'm talking about, right? Like, we should have everyone's face on so I can, at least people's nodding. You know, where the bees are like, yeah, they're, in this case, technically in this case, they were bees with a color dot that landed at the bottom board and tried to get into the entrance. Whether they got into the entrance or not, we didn't know, and we never found a colored bee in those colonies. But none of these colonies experienced massive robbing, so it wasn't a robbing event, but they were clearly being visited by these bees.
They're nosy neighbors.
Yeah.
Maybe they're scouts. Maybe they're checking things out.
Yeah, but they never actually initiated that robbing behavior. Yeah.
I think everyone's in agreement. We like snooping behavior as a term, so. All right, everyone. So do we have any other questions for the speaker? If so, feel free to ask them now or drop a question in the chat. And, okay, last call. And I think we will wrap it up. But, oh wait, no, never mind. I saw someone on mute for a second.
I did. I was going to ask Dennis if the Spanish group was looking for more mite samples and if we could, you know, have more of the mite resistance and markers looked at in regions that haven't been studied yet.
Right. So I think that, I mean, I think it was a pretty exhaustive with this fluvalinate. I think that we have sent a lot of samples. When people ask for mite samples, we're pretty generous with them. So we've sent some to Japan. There's a big group there that's doing a lot of really deep sequencing now. And certainly we share them with anyone who needs them. I think one of the questions, and this might be a good topic for your AI meeting, and it'd be interesting to discuss, should we also be seeing, well, if we're doing some of this work with viruses, should we be trying to do a layer where we're also doing genetic resistance assays as we go, so that there's some, so we're tracking that too. Like that's a way of thinking about this too. Should we be including that as a standard part? And certainly it is pretty simple. It does require a different step because I think it requires DNA extraction rather than RNA extraction. But I think that's true, right, Judy? Yeah, you can use the RNA extracts.
Yeah, and how many are looking at amitraz resistance? Is it just singular focus on one compound, or is there a broad range of compounds that you're looking for a resistance marker?
I think they haven't been able to find many. They haven't been able to find the amitraz markers yet for resistance. I don't think anyone's actually found for sure a resistant population of mites that are amitraz resistant. Have they? I mean, I think it's been hard to make sure that they are resistant after several tests.
I think it's anecdotal. There's a lot of beekeepers that claim resistance,
Yeah.
but the researchers are having difficulty isolating them.
Yeah, and every time people send us resistant samples and we do it in the PETIS test, we find it successful. And Christina sometimes finds there's a variability there, but we have never found real resistance. So that makes it hard then to look for a gene that's causing it. So the number of markers we have isn't, but I know that there are a lot of groups pursuing that. I think that the approach now people are taking is the whole genome approach, where they're trying to sequence the whole genome and see what's happening.
Emily Wine asks, some miticides such as formic acid kill brood. Would this affect the percent of mites? If both bee and mite populations decrease, the mite load may not decrease.
Yeah, so that's a really good question, Emily. And I think that that's actually one of the reasons sometimes we see strange mite populations is if there's a sudden loss of the forager force, we can see spikes in the mite to bee ratio. You are right too that it can kill uncapped brood. Typically though, when I was doing this work with formic acid pads, just as that was all developing, I remember clearly that if we killed brood and we would measure that brood kill, we would find that they would make up more than enough from it afterwards. And so the population rebounded. But you're right, that's a really important point. I think that's underlying your question, is that are some of these bad effectiveness, the poor effectiveness of our product simply a function that we're not able to really quantify the mites in the colony in a way that's efficient. And I know that there are several people who've been thinking about, Marla is thinking about this too. I think, how do we measure mite populations and success? Do we actually need to be getting under the brood and all these other things? And that's been a question for as long as we've studied Varroa mite, right? The sticky board versus the sugar roll versus the brood capping. And there are advantages and disadvantages of each. And certainly there's the information we get from just doing adult bee counts is limited.
Thank you very much. All right, so I think that will wrap it up today. But if you have any questions about after the talk, I know sometimes it's hard for me to think of things right on the spot, but I think about it later. If you have any questions, you think about it tonight, feel free to send me an email and I will forward that on to Dennis. So yeah, thank you so much. This was great, Dennis. I really appreciate you.
Thank you. Thank you, everybody.
All right, thank you.